Now look at this.
What's up, about penguins? Today we're going to do topic 2.5 on membrane permeability. So as a reminder, the plasma membrane is made up of these phospholipids, and they associate into a way where we have the hydrophilic heads facing the interior of the cell and the exterior of the cell, and hydrophobic tails facing the interior of the membrane. And so what this does is it causes the membrane to be nonpolar. So the items that are able to pass through the membrane easily would be things that are small and nonpolar. So like carbon dioxide, oxygen, those are items that can just easily straight up pass through. We oftentimes talk about like steroids, like testosterone and estrogen. Those can pass directly through the membrane because steroids, we know, are nonpolar. Because, in fact, the interior of it is nonpolar, we are going to need some type of transport protein in order to move things that are polar. So like charged particles like sodium and potassium, or like water would require a transport protein in order to pass through it. Fun fact. Water is able to slowly slide through the membrane, so it can leak, but for the most part, water is going to be through a transport protein. It's important to note that these channel and carrier proteins that transport materials across the membrane are specific for the ion that they transport. So if we're talking about like a sodium channel, it is only going to bind to sodium due to size constrictions and due to the way that it binds to that ion. And so remember we talked about that proteins have active sites, and so it's going to bind to the active site on that protein to allow it to pass through that membrane. So they are specific for what they travel through. And if material is way too big to pass through that membrane, then we oftentimes look at like bulk transport. So like phagocytosis and pinocytosis and exocytosis would be how we're going to allow some of those bigger molecules to move across that membrane. So a couple different organisms are going to have cell walls. So we talk about like plant cells have a cell wall, fungi has a cell wall, as well as certain bacteria have cell walls. So plant cells are going to have cellulose within their cell wall that provides that rigidity and that strength to the plant cells, as well as notice that it has that central vacuole. We've talked about this before. The central vacuole is going to provide a force when we're in a hypotonic environment to allow our cell to stay turgid or erect when we think about like our plant cells. They want to be turgid. They want to be in a hypotonic environment. That's what is favorable for plant cells. Fungi are going to have the polysaccharide of chitin in their cell wall. And then some bacteria have a cell wall with peptidoglycan in it. That's how we tell the difference between a gram-positive and a gram-negative bacteria, is if it's gram-positive, it has peptidoglycan that takes up the crystal violet, turning purple. Don't worry about gram-positive and gram-negative, though. Remember, I'm doing this for all the units, so be sure to hit that follow button. Remember, AP Penguins, dress success. Bye, y'all.